Hi, my name is Hannah Padre, and I'm going to be talking about the conserved region known as Carmel Sea. This is a quick overview of my presentation. First, I'm going to provide background on the protein that I chose and its conserved domains, and then I will go into my method to finding sequences from other organisms, and lastly, I'm going to go over my three multiple sequence alignments. The first two multiple sequence alignments help me create my amino acid sequence motif, and then the last multiple sequence alignment, it gives me a phylogenetic tree that will show the relationship between the different organisms. And when I get there, I'll explain why I only chose to perform multiple sequence alignment on only Carmel 1 proteins. The protein that I chose is actually Carmel 2. It has a much longer name, but I'll just call it Carmel 2 for short because that's the gene that encodes this protein. And so these are screenshots from the NCBI website that gives me more information about the protein, such as the length, which is about 1,435 amino acids long, the accession and version numbers, and also a summary. So just a quick summary about the protein. Carmel 2 is involved in the regulation of actin polymerization at the barbed end of actin filaments. Essentially, this protein negatively regulates another protein known as CAPZA2, which regulates the ARP23 complex's polymerization activity. So essentially, the presence of Carmel 2 increases polymerization. And while on the NCBI webpage, if you select Identify Conserved Regions, it brings you to the Conserved Domain Database, CDD for short. Here we have several conserved domains, but my focus is going to be on Carmel C. So Carmel C is found near to the C-terminus, leucine-rich repeat-containing proteins in the Carmel family. And in LRRC16A, it includes a region responsible for interaction with the CAPZA2 protein, which is the protein that I mentioned earlier. So after choosing my curated protein, I can begin my search for other organisms that share the Carmel C region. So I select Identify Conserved Domains again, and then I select Carmel C. And then I select specific protein because this retrieves protein database records that have a high degree of similarity to the conserved domain. And then I filter my results to only return RefSeq sequences. I noticed the first result is not the same protein as the one that I chose. So before searching for other organisms, I decided to take a look at what other proteins in humans contain Carmel C as a domain. So first I filtered down to only Homo sapiens, and then I sorted by accession. I sorted by accession because it returns curated sequences first, so sequences that begin with NP. This is the result for my first round of filtering. And as you can see at the top, that's the protein that I chose. And below that is actually a protein known as Carmel 1. And at the bottom, that's a protein known as Carmel 3. Oh, sorry. So these are all different proteins, but they contain the Carmel C region, and they're all a part of the Carmel family of proteins. I save all three files, the sequences, into a text file for my multiple sequence alignment, and I begin selecting other sequences. And I just did this manually. I filtered by organism, selected a few from mouse, selected a few from cows. And in total, I selected 11 sequences. Like for example, I chose 2 sequences from mouse, but one is for the Carmel 1 protein, and the second one is for the Carmel 3 protein. And to the left, I included the shorter name for the long name, such as F-actin capping protein is Carmel 1. And so copying and pasting everything to my text file, I also changed the sequence names to the organism first, and then the protein, and then the accession number. So this is my resulting multiple sequence alignment. It's actually really big, so I'm just going to show you the regions with long stretches of conserved regions. So like for example, we have from 178 to 382, and we have 654 to 676. But when I look back at my curated protein and the region for Carmel C, it's actually between the stretch of 800 to 1,000. And as we can see, there aren't too many conserved regions as I was expecting. Now, taking a look at percent identities, I noticed that when they share the same organism but different protein, such as Carmel 1 and Carmel 2 for both humans, it has a low percent identity. But when it's the same protein and different organism, like cow caramel 1 and human caramel 1, it has a higher percent identity. So I guess this makes sense because the same proteins should technically be more similar. And then taking a look at the phylogenetic tree is a bit interesting because it clusters the sequences by other protein groups and not by organism. I guess I should have included the guide tree, but this will do. So as you can see here, all the caramel 3 sequences are grouped together, and then caramel 1, and then caramel 2, and LRR. So I trimmed down all my sequences to the regions that only contain caramel C for my second multiple sequence alignment. So I did this by identifying the region, trimming down the region, copying and pasting the sequences to a text file, and then renaming them to organism first, the protein type, and the accession number. And these are my results for my second multiple sequence alignment. So I got similar results to my first MSA. There aren't any long stretches of conserved regions, but there are still conserved amino acids here and there. Looking at percent identity, it's actually lower. There are a lot lower numbers than my first one. And phylogenetic tree, I could kind of infer the same thing, like from the first one that we saw. Oh yeah, and so before I could show you that slide, I took these sequences up here that are in the green bar or below the green bar, and I entered that into WebLogo to create a sequence motif. And yeah, so we can see, there's some amino acids that really stand out. And to compare, I also found the PFAM database sequence or logo motifs. I found that from first starting at the Caramel C CDD page, selecting PFAM, and then selecting HMM logo, and then I put them side by side. So as we can see, there are some similarities circled in black, and there's a lot of other differences. But yeah, we can see that there are still some really prominent amino acids, such as the CP, cytosine, and proline, which are both nonpolar. So I guess we could infer from our own logo that there is highly conserved amino acids that have some sort of structural or functional importance to the caramel C domain in the protein. It's really cool to see this from different proteins and not only using caramel 2. And it's cool to see that my web logo is pretty similar to the one on the PFAM database. So lastly, I decided to create a third multiple sequence alignment, but only with my caramel 1 proteins. And I just chose caramel 1 because after collecting, that was most of them were caramel 1. So trimming down to the regions and entering them into Cluster Omega. This is my resulting MSA. And as expected, the MSA shows long stretches of similar conserved regions. My percent identity has high values. And now we can take a look at the phylogenetic tree. So cow and human are clustered together, the birds are clustered together, and the mouse has its own branch. And by looking at the percent identity matrix, we can see that the crow and fowl have the highest percent identity and are most closely related. And crow and mouse are most distantly related. And yeah, that was my presentation. So just some final thoughts. I was surprised with my first 2 multiple sequence alignments. Like I thought there would be higher percent identities, and I thought my conserved domain would contain more conserved regions. Although I was glad to see my web logo was so similar to that of the PFAM database. And it was interesting to see the amino acids that are so highly conserved among different proteins. I also found that to see the phylogenetic relationship between different organisms, or the evolutionary relationship between different organisms, it's better to use the same protein, like caramel 1. So yeah, that concludes our presentation. Thank you for watching.